perhaps. I'm a lecturer at the University of Malta. I'm a medical doctor by training, but then I decided to take up research at the university, and I went also obviously to universities abroad, and I'll mention about that a bit later on. And my area of study is in fact that of Parkinson's disease and also Alzheimer's disease. Okay, so I've started doing this type of research back in roughly 2008, after I finished my PhD. And I started a research group, a research team, with the aim, so we work in a laboratory, we don't work with patients, we don't work with patients, we work in a laboratory. And we try to answer key important questions using science. That's why the title of this talk is The Science of Parkinson's Disease. So we try to understand things like what is causing Parkinson's disease, why do nerve cells die, and how do they die, and most importantly, to use this knowledge then to develop new treatment. So once the idea is, once you have the knowledge of what's happening in the brain, you can then devise treatment in order to stop it and to prevent it. And during this talk, I'm going to, obviously it's not an easy talk in the sense that science, it won't be easy to explain to. However, I'll do my best. Okay, I'll do my best. I'll try to keep things as simple as possible, so that you get an idea of this field of what we're doing, and the state of research on Parkinson's disease. So the idea is as I said, to showcase what we're doing, and the ultimate aim is to develop some form of new treatment. As you know, the treatment that exists now, and which some of you may be already having, is what we call symptomatic treatment. It improves the symptoms, it's effective in improving the symptoms. However, unfortunately, it does not address the cause. So it does not stop the disease from progressing. So we need an effective treatment, a more effective treatment, to actually stop or decrease, slow down the progression of the disease. And that is the aim of our research. So perhaps I can try to share my screen now. But you have to allow me, Cassandra, to share host disabled participants. So you have to allow me.
That it worked.
Where did you go?
You have to make me as a co-host or something like that.
Okay, it should be okay now.
Okay, Mala, share screen. That's it. Very good. Okay, so there you can see, you should be seeing my title page. Let's start the presentation. That's it. Okay, so are you seeing the flowers there and the other strange objects? Very good.
Yes.
Okay, very good. As well, the pointer, are you seeing the pointer moving?
Yes.
Very good. Okay, so I can show you. All right. So I decided to put the tulips. There, the tulips are the symbol of the Parkinson's disease and research on Parkinson's. So the tulips in the middle, and some things that are going on in the brain, which I will be explaining very soon. Okay, so first slide is going to take us 200 years ago, more or less, 1817. Okay, so this is where the whole story about Parkinson's disease started. In fact, there was this English physician or surgeon called James Parkinson, who published in 1817 the first description of a person with Parkinson's disease. He called it an essay on the shaking palsy, shaking because he noted that the tremor in the hands. So this was the first. In fact, you can see this is a copy of that first publication, an essay on the shaking palsy by James Parkinson. So he noted that there are this condition in which people would have slowness of movement, together with a tremor, perhaps a stooped, what we call a stooped posture. He described it. So he described it for the first time, and in fact it's named after him. So that's why you call it Parkinson's disease, after this English physician whose surname was in fact Parkinson. The next big advance was almost 100 years later, and now we have to go to Germany. So in Munich, in Germany, around 1913, so from 1817 we went to 1913, there were a group of doctors who were studying the brains. Now they were studying the brains of people with Alzheimer's disease and Parkinson's disease. In fact, here in this is in this group photo, we can see this is the guy. He was called Friedrich Heinrich Lewy. As his name implies, he was Jewish in origin, Lewy. And this person here in the middle, this was Alzheimer, which gave rise to the disease which is known as Alzheimer's disease. So actually Alzheimer's and Parkinson's disease, they were linked from the very beginning. The two doctors who described Alzheimer described the first patient with Parkinson's disease. Lewy, what did Lewy do? Lewy, he first identified in the brains of people with Parkinson's disease, he identified, and these are some of his actual drawings. These are some of his actual drawings. He drew these brown-shaped, they call them bodies in English, bodies. But he saw them in the nerve cells. In fact, they were called after him, Lewy Bodies. Lewy instead of Lewy, because he had to emigrate due to the when Hitler came in Germany. He had to leave Germany, and he went to the United States, and he changed his surname from Lewy to Lewy. In fact, he died there as Frederick Lewy. He died in 1950 in the USA. So, and we call them still today. We refer to these as Lewy bodies. So these are going to be very important for our talk, these Lewy bodies. So these are basically plaques or aggregates which start appearing in certain neurons, certain nerve cells inside the brains of people with Parkinson's disease. So this was the discovery of Lewy, and incidentally, this was at the University of Munich, and we're going to mention Munich again a bit later on. I'll tell you, then you'll see why. Okay, now a bit further on. So now we go to 1960. So more recently, another major discovery on Parkinson's disease was the discovery again by another German group of doctors. The discovery of the association between Parkinson's disease and the chemical dopamine. They found that there is a lack of this chemical dopamine in certain parts of the brain. We call these the striata in patients with Parkinson's disease, striatal dopamine deficiency. All right, and this perhaps some of you in fact may know this, because this is usually what we tell patients who tell them that there is a deficiency of this chemical. In fact, again, one of the main treatments which exists for Parkinson's disease is in fact to give tablets that have dopa, which is dopamine. So we replace this chemical which starts lacking in the brain. And this chemical is important for movement. Okay, this chemical is important for movement. So this was discovered around 1960. So, and I have here this sort of this image of the dopamine molecule. Okay, so we have these facts. So we have the Lewy body. So we know from the work that Lewy did that there are these Lewy bodies formed in the neurons, and we know that there is a deficiency in dopamine. In fact, I mean, this is a modern picture, okay, which is showing you. It's called a PET scan, which shows the site exactly where there is the loss of this brain chemical dopamine. Okay, so it's, as you can see, they are located sort of towards the base of the brain. They are called the basal ganglia. They are called the basal ganglia. And on this PET scan, you can see this is showing you the red means a lot, means there's a lot of dopamine. So this would be a healthy patient, a person. And here we have a person with Parkinson's disease. You have this. It's less. The color is green and yellow, meaning confirming that there is a deficiency of dopamine. So the question arises now: why is one of the most important questions that we have to ask? Okay, there is this lack of this chemical, but why is this chemical becoming deficient? Why are the neurons? Why is there a loss of this dopamine, a lack of this dopamine in the brain? We know that there is a lack of dopamine in the brain because the neurons that make this dopamine in the brain, which are located here, okay, the neurons, the nerve cells which synthesize this dopamine start to die. Okay, so there is loss of cells that make dopamine in the brain. Okay, and the cells that make dopamine in the brain are found in a place called the substantia nigra. But anyhow, that's not so. Okay, so there is loss of cells that make dopamine in the substantia nigra. As a result of the lack of dopamine, there's going to be the problem with movement. So the question that arises now is, why are these cells dying? Why are these cells that make dopamine in the brain dying? Okay, what is happening? What is causing them? What is causing them to die, and therefore not to keep on making dopamine? And here is where we go back to our Lewy bodies. Okay, so when they looked at the microscope, when they looked at these neurons that were dying in the microscope, you can see on the left side that is a normal dopamine inside the cells. On the right side, you see cells without dopamine. Okay, when they looked at these cells which were dying, they found that they were filled with these Lewy bodies that Lewy had described many years earlier. So the link was the scientists, the medical people, they started thinking, could it be that the Lewy bodies contain something which is killing, which is causing these neurons to die? Could these Lewy bodies, which are forming inside these neurons, okay, because these Lewy bodies, they are found, that's one example here, that is one, this is another one here, they are round, this is another one, you can see that's, okay, so this is another one, okay, so these are the Lewy bodies that you saw when he looked at the brain, all right, and now we know that these form in the places in the neurons which make dopamine. Okay, so an obvious conclusion is that, listen, then these Lewy bodies must be doing something bad to the neurons. What is that? How are they killing the neurons? So that is the next important question. So now we come to even a more recent date, 1997, all right? When there was this very important discovery by Maria Grazia Spillantini, an Italian working in Cambridge, in the University of Cambridge in the United Kingdom. Maria Grazia Spillantini. In fact, this is the paper that they published in the study they published in an important scientific journal, in which they found that those Lewy bodies that Lewy had described practically 200 years before are made up of a protein called alpha-synuclein. So they looked at them with a very, very, very strong microscope, and they found that these Lewy bodies, these round Lewy bodies, on a very strong microscope, they appear like this. Huh? They appear like this, like threads, Actually, they represent clumps, aggregates of this alpha-synuclein protein. All right. So essentially, what is happening is that these Lewy bodies are formed because of clumping, abnormal clumping of this alpha-synuclein protein. In fact, in the same year, there was another study which found a large family in Greece, who had, and this family, there were many, many cases of Parkinson's disease in this family. Okay, so brothers, aunts, uncles, there were many, many cases of, and they found that in this family, they had a mutation in the gene coding of alpha-synuclein. Okay, so they linked the reason for the large number of cases in this family of Parkinson in Greece was actually because they had alpha-synuclein, and therefore, because they had this alpha-synuclein, they were having a lot of Lewy bodies. Because they had a lot of Lewy bodies, the neurons were dying in the substantia nigra, which make the dopamine, and therefore there was a lack of dopamine, and therefore there was a lack of this chemical, and hence the motor problems. All right. So we are sort of tracing the story, starting from the patient, then the brain, the Lewy bodies, the dopamine, the alpha-synuclein now. So now we are really targeting. So the importance of this research is that, okay, so alpha-synuclein is important, that we try to stop this alpha-synuclein from aggregating. Okay, so this is a summary of what is happening. So we have these alpha-synuclein, this protein, which is forming first, they start sticking, so this is happening inside the neurons which make the dopamine. This is happening inside the neurons which make the dopamine. So this alpha-synuclein, they start sticking to each other. Then they form those clumps, those strands, all right? And finally, they form the Lewy bodies, which we can see under the microscope, under a normal microscope. To see this, you need a very special, you need an electron microscope, a very special microscope, a very strong microscope to see these. To see the Lewy bodies, you need a normal microscope. And so the conclusion was that, okay, Mela, it's these alpha-synuclein clumps, aggregates in English, clumps, which are causing death of the nerve cells which are making the dopamine. All right. So again, the next step now is perhaps how do we stop these clumps from forming? Because that is the bad thing. How do we stop the clumps from forming? And another question is as well, why is the neuron dying? Okay, we know that it is dying because of these clumps, but how exactly are these alpha-synuclein clumps causing the nerve cell to die? All right, and here I'm showing you a bit of a small animation, a short animation to show you how this clumping occurs. So this would be an alpha-synuclein protein inside the cell, and you can see basically it starts to stick with others. That's why I told you they form aggregates, they form clumps, and then these clumps, they form these long fibrils. We call them fibrils, and you see that is the Lewy body. Okay, let me show it to you again. all right? So that would be the alpha synuclein. They would start clumping, sticking together. You can see that they're sticking together, and then they are forming these larger and larger aggregates till you finally get to the, so this is sort of an image of a Lewy body forming inside the cell, and then somehow this is going to cause the cell which makes the dopamine to die, and therefore there's going to be a deficiency, a lack of dopamine, and hence the Parkinson's disease. Okay. So again, this is another image showing all those orange dots would be the aggregates, the clumps which are forming in a neuron which makes dopamine inside the brain, in that part of the brain which makes dopamine. Okay, so now we come perhaps to our own research. Okay, so in 2008, as I was telling you, we started the laboratory doing work on neurodegenerative diseases, which includes Parkinson's and Alzheimer's disease mainly. I'm going to tell you a bit about our research and the questions that we answered. Okay, so one question that we wanted to answer is how could these clumps be causing death of the neurons? All right, so okay, we know that they form inside the cell, but exactly how are they killing the cell? Why does the cell die? And we shifted our attention to structures inside the cell called mitochondria. So perhaps those of you who have a bit of a biology know mitochondria, what mitochondria are, but essentially mitochondria provide energy to the nerve cell. They are, that's why I put it, this is a power station. Power stations provide energy to the country. Mitochondria, you can see these here, 1, 2, 3, 4, 5, 6, 7, these are all mitochondria. Mitochondria, they provide energy to the neuron. So our question was, could it be that those aggregates of alpha synuclein, all right, are causing damage to the mitochondria so that then the cell dies? Because obviously without energy, the cell is going to die. The brain cell is going to die without energy. So could it be that it is causing damage to these mitochondria one by one, causing the cell to lose its energy and therefore die and therefore not make the dopamine? So that is the question that we asked. And so these are actual. This is an actual picture of cells, of brain cells in the lab. As I told you, we don't do our research. We don't do research on people. We do research on cells and alpha synuclein, etc. in the lab. So in the laboratory, we replicate what is happening inside the brain of a person we try to replicate, let's put it this way, we try to replicate what is happening in the brain of a person with Parkinson's disease. Right? So this is a photo of cells, of brain cells that we grow inside the lab. So we grow these cells inside the lab, and we studied whether, so we made clumps of alpha synuclein as well. We made clumps of alpha synuclein, which again you find in the brain of patients with Parkinson's disease, and we looked at what's happening to the mitochondria. So this is a large mitochondrion, and we tried to see if can the alpha synuclein cause damage to the mitochondria. So these represent the clumps of the alpha synuclein. Can they be causing damage to the mitochondria? In fact, we found that it did. So that was one important finding. Mitochondria are badly damaged. So when we found that this alpha-synuclein, the clumps inside the cell, do great damage, big damage to the mitochondria. And therefore, in fact, this is another photo. This is a normal mitochondrium, and this is an abnormal mitochondria. It's swollen, and it has holes in it. In fact, we found that these alpha-synuclein aggregates, they punch tiny holes in the mitochondrion. They punch small holes in the mitochondrion and basically causing it to they destroy the mitochondria. They destroy the power stations. Okay, so the cell ends up without energy. In fact, you can find this. We published this study recently in 2019, 2 years ago. And you can find this on the internet. In fact, okay, so we published it in a journal of the American Chemical Society. Okay, and we collaborated, in fact, with a group in Germany. At the Center for Neuropathology of the Ludwig Maximilians University of Munich. Incidentally, this is the same university where Lewy had done his studies on the Lewy bodies back in the early 1900s. So it's actually this university has a long tradition of study on Parkinson's disease. Okay, so we did the study together with this university in Germany. Okay, and in fact, okay, just to show you, this is not our lab. I wish it was our lab, but it is not. This is the lab. This is the it's a huge structure, right? And at the university in Germany, dedicated just to research on Parkinson's disease and Alzheimer's disease. And you can see the rows of labs, of benches of labs inside this building. Okay, so this we're going to continue this story a bit more now, because we come to a very interesting part. So in 2013, this guy here, Professor Armin Giese in the University of Munich, and in fact there's a photo of me at that university, because I carried out part of my studies, my PhD, in fact, at this university as well. So it's a beautiful city, Munich. It has tradition. It is cosmopolitan. And it is very strong in research. Okay, so they have a very good university hospital there, and they are very well funded. The research is, as you know, Germany. They have very well funded universities. Anyhow, so in 2013, the Professor Giese discovered a compound which they called One Triad B, which, and here is basically a bottle of this compound, okay, which they found was very effective at preventing the aggregates from forming. Okay, so the idea was to try to find a compound, to try to find a chemical, a medicine, which is able to stop those clumps from forming. Because obviously we knew that it was those clumps which are causing the neurons to die. And they screened. Therefore, the idea was, if we can stop these clumps from forming, if we can prevent the alpha-synuclein from aggregating, we can therefore ensure the survival of the dopamine neurons. So the dopamine neurons, they won't die. The dopamine is going to keep on being produced, and we delay or we actually stop the Parkinson's disease from progressing. So this is the ultimate form of the best treatment that you can get. all right, in a way. So they tested 20,000 compounds, 20,000 compounds. And here you can see some of them, 1, 2, 3, 4, 5, 6. These are all different compounds, different chemicals that they tested. All right. And here, whoop, this is another list from another page. 1, 2, 3, 4, 5. Okay, 20,000 of compounds. And they tested, they looked at how effective each compound was in preventing the alpha synuclein from aggregating. That is what they wanted to see. They wanted to see the most effective, the best compounds which prevented the alpha synuclein from aggregating. And finally, they identified one, this one, only 138B, okay, which was one of the best compounds, and that is only 138B. So this compound, this possible new medicine, okay, for Parkinson's disease, is able to prevent those clumps from forming, and from what we know, therefore, it prevents damage to the mitochondria, to the power stations, and therefore the nerve cells do not die, and therefore the dopamine remains being produced. Just to show you some results, okay, and from this research, here on the left-hand side, those yellow dots, they all represent the alpha synuclein aggregates. Each dot represents which we found in the Lewy bodies. So these are more or less the Lewy bodies. Let's put it this way. On the right-hand side, they tried the same thing. They looked at what's happening in the lab, okay, with only 138B present, this medicine. And they found that when this medicine was present, you can see there is much less of these aggregates, of the alpha synuclein aggregates, which form the Lewy bodies. There is much, you can obviously see it with your own eyes. You don't need to be a scientist to see that. Okay, so this is the control. This is the Parkinson state, and this is with the only 138B. So this is showing that it very strongly prevents the alpha synuclein from aggregating. This is in the lab. Now this is in a test tube, sort of. They also did studies in mice to move more closer to humans. Okay, so they did studies in mice, and one of the studies in mice that they did was, it's called the Rotarot performance. I'll show you a bit. So basically, those are mice there. The mice are put on a rotating rod. They are placed on a rotating rod, and at one point, the mice, they are going to fall down. So here you see this guy here. All right, after a certain time, is going to fall down, and then they studied the mice that had the Parkinson's disease. Okay, so you see, it fell down. All right. They studied the mice that had Parkinson's disease. They would fall more often. The mice that had Parkinson's disease. You tell me, how do the mice have Parkinson's disease? Basically, they get Parkinson's disease by giving them alpha synuclein. So they give them alpha synuclein. The Lewy bodies form in the brain of the mice, and the mice get a form of Parkinson's disease. Not exactly like the human one, but essentially they have problems with movement as well. And one of the problems with movement is their ability to stay on top of the rod. All right, to stay on top of the rod. So the mice that were given the alpha, they dropped more frequently. In fact, they dropped twice as much. Okay, so the healthy mice dropped 15 times. For instance, the mice with the Parkinson's disease, they dropped 30 times. Now, the interesting thing is, when the mice were fed, they were given only 138B. When they were given this compound, okay, when they were given this compound as a medicine, okay, the mice, they started falling less. Okay, the mice, they started falling less. You can see, almost back to 15. So this is showing us, this showed them that this is effective treatment. This treatment, the One Triad, improves the motor performance of the mice. Okay, so not only does it decrease the alpha synuclein aggregates, but it actually improves the motor performance, and this is very, very important. They also found that it improved the survival of the mice. All right, so mice which had Parkinson's disease, they lived up to, say, 550 days, more or less. All right, the mice that had Parkinson's disease and also were given this treatment, the One Triad, they survived more. They survived around 100 days more. Now, 100 days for a mice is equivalent to like between 5 and 10 years for a human being. Okay, so they improved the survival of the mice. So this is very important. This is very important research. Okay, this is very important research. We're showing that this compound can stop the aggregates forming in the brain. It can improve the performance, the motor performance, and it can increase survival. Okay, you tell me, but this is a mice. It's true, but that is a very important step before you go on to humans. All right. Again, so this is another slide showing how, in the presence of only One Triad, these are the Lewy bodies, the alpha synuclein. With only One Triad, there is practically no brown. You can see no brown here. Okay, so that shows you a lack of the alpha synuclein aggregates. Something that we did with this compound research, that, as I told you, we're very fortunate to be able to collaborate with this German group on this research. We found that in our laboratories at the university, that the mitochondria, remember we said that the alpha synuclein damages the power stations, the mitochondria. We found that the NLI prevents the damage to the mitochondria as well. Okay, so this compound, this medicine, prevents damage to the mitochondria. And the mitochondria remain healthy. So the mitochondria remain healthy, the cells do not die, therefore. Once the mitochondria are healthy, the cells do not die, and dopamine is going to keep being produced. Right? So this is important research. In fact, again, we published this research in 2017 together with the German group. Coming to the humans now, obviously before you can market a drug to be used in patients, you have to do clinical trials. We've heard about clinical trials with the COVID vaccine, which they told us that clinical trials usually take many years, and in fact they do. And it's actually a miracle that we have a vaccine in 1 year. Clinical trials, you have to do them for any medicine, any new medicine has to pass through what we call clinical trials. All right, so we have to have evidence that it works in humans, evidence that it works in humans, and evidence before that it is safe. So in August of last year, in fact, okay, this compound, One Triad, was given to people for the first time. It's called the phase 1 trial. There are three. There's phase 1, phase 2, and phase 3. Phase 3 would be, so you start with phase 1 trial, then you go into phase 2, and then you go into phase 3. If your compound passes all these, it passes all these tests, then finally you have chance to actually give the medicine to patients. In the phase 1 trial, so in August of last year, there was completion, it finished. The phase 1 trial was successful, so they gave only One Triad up to 300 milligrams daily. So they gave them a dose of only 1,3,8-B to people. And they found to healthy people, that is how you start. You don't start on patients first. You start on healthy people. So first you see that it doesn't do any harm. Yeah? In medicine, we say primum non nocere, something like that in Latin, coming from Latin. Primum non nocere, first do no harm. Prima non nocere, first do no harm. That's an important principle. Otherwise, don't do anything. So this drug was tested, and it demonstrated, it showed excellent safety in doses of up to 300 milligrams daily. So we know that we can give this treatment now at up to 300 milligrams daily. In December of last year, in the midst of the pandemic, I would say, there was initiation of the phase of the second step of trials now, in which only 1,3,8-B is being given to patients with mild to moderate Parkinson's disease. So that would be a lot of patients. Okay, so patients with mild to moderate Parkinson's. This study is in fact being conducted at the moment at the neurology department of Nottingham University Hospital. So together with the in the United Kingdom, in fact. Okay, so now the drug is being given actually to patients who have mild to moderate Parkinson's disease, with the idea at this stage to confirm that it is safe in the patients. Safe in the sense that it does not do damage, for instance, to the liver, it doesn't do damage to the kidneys, it doesn't cause any allergies, it reaches the brain. So one important thing, for instance, that this compound, which you take by mouth, into the drug, you take by mouth, which goes into the intestines, which then goes into the blood, it has to reach the neurons in the brain. It has to reach the neurons in the brain, and that is not easy. Okay, so you need to find the right dose so that the compound reaches the brain. So we're in this stage now, where the phase 1b trials have started with this compound in order to determine the safety and tolerability of only 1,3,8-B in PD patients. The next step, so if they find that this treatment is safe to PD patients, the next step would be then to try to see whether it actually is able to improve the symptoms of Parkinson's disease in a small group of patients. And if that succeeded, then in the phase 3 trial, they would try it on a larger group of patients. Obviously, this is going to take several years, but till now, things are looking quite good. Perhaps a note here is that in fact this compound is also being the research. Obviously, research costs a lot of money. In fact, this research on only 1,3,8-B is being funded also by the Michael J. Fox Foundation in the United States for Parkinson's research. The Michael J. Fox Foundation, as you know, perhaps Michael J. Fox is the famous actor who developed Parkinson's disease. And the money he donates millions. He donates millions of dollars every year to support research around the world on Parkinson's disease. Okay, so and in fact, these trials with only 1,3,8-B are in fact partly funded by the Michael J. Fox Foundation. Obviously, around the world, many people are trying many different compounds. Okay, unfortunately, a lot of compounds fail when they come to the phase 3 trials. I told the phase 1, phase 2, phase 3. So here is an example of different compounds. I won't go into the details, but just to give you an idea, in which they found that they stopped at phase 3. So in phase 1, they were safe. in phase 2, they looked promising, but then they failed in phase 3. Okay, so obviously we hope that this won't happen with this compound, but the reality is that it's not easy to find effective treatments in large clinical trials. Okay, but the hope is there. The hope is there. Again, the slide, I won't go into this, but just to tell you that only One Triad is one compound. All right, there are many compounds. These are in gray. There are many compounds which are being tested. Okay, so it's not just around the world in order to try to find new neuroprotective therapies which are being tested in clinical trials. So this is to emphasize really that there is a lot of research going on Parkinson's disease at the moment in order to try to find a treatment which stops, as I told you in the beginning, stops the progression of the disease. One last, actually, okay, it's all right. I've been 45 minutes talking, so I won't. But I wanted to mention this. Another aspect of another thing that we found with the research is on natural compounds. And this is interesting as well, perhaps for another time. We studied chemicals which you find in berries and fruits and strawberries and tea. And we found that these compounds were able to break down the clumps of alpha-synuclein as well. And this is really, really interesting. So this is a healthy diet now. So this is not medicine. This is and we published this. In fact, also we published this research. You can find it on the internet as well. So this is really, as we found, especially black tea, which was quite effective. And in fact, here I'm just going to show you quickly a study that we did by black tea. Okay, so you see those are the aggregates of alpha-synuclein, and when we added the black tea compound, you can see there was much less. There were much less aggregates. Okay, so this is another area of our research. So which is to look at compounds in and I know that Cassandra as well is interested in this. Yes, healthy eating. And in fact, at the time that we were doing this research, in 2012, in fact, an important study came out from America by Harvard University. Harvard University is the top university in the world, basically. Harvard University Medical School, in which they studied 130,000 men and women for 20 years. So this was really a massive study, which took 20 years. So they finished it, they started it in 1992. And they found, they looked at how many people got Parkinson's disease, how many people didn't get Parkinson's disease, and they tried to link it with their diet, And they found that men, this was significant in men, that men who ate flavonoid-rich foods, flavonoid-rich, meaning the healthy food like berry, fruits, tea, apples, and red wine. Okay, flavonoid significantly reduced the risk of developing Parkinson's disease up to 40%. That's a very strong decreased risk. Okay, so this is not treatment, this is prevention of Parkinson's disease. So they found that those men that had a healthy diet, with more fruits, berry fruits, tea, apples, and red wine, they were protected against developing Parkinson's disease. And this linked very nicely with what we were seeing with our compounds, which with the studies that we were doing in the lab with our compounds on strawberries, berries, etc., and especially tea, as I mentioned. Okay, so to conclude, and I'll stop here. All right. So first of all, perhaps one message that I want is the importance of alpha-synuclein. Okay, so the disease is being caused by this alpha-synuclein, and we are trying to find basically new medicines which attack this alpha-synuclein. And our hope is that this new compound, One Triad B, might be really, it's showing that it is effective in the laboratory. It showed that it is effective in mice. We're hoping that it is also will be also effective in humans to provide a new form of treatment. So thank you very much for your attention and for bearing with me through this perhaps not so easy talk. And I also obviously would like to thank the my many students who did this. Obviously, I don't do the research all myself. So the many students who came to the lab, who studied and did masters and PhDs with me, and who did a lot of the research that I've been talking to you about. Okay, thank you. So that brings me to the end of my talk, Cassandra.
Amazing. All right.
Thank you.
Thank you very much.
Thank you very much.
Well done. Does anybody have any questions?
Yes. The floor is open.
Professor Vassallo.